Hello students, welcome. Let's get started on our lesson on how to study organisms. By the end of this lesson, students should be able to give the simple safety precautions to follow when observing organisms. Organisms can be observed in their natural environment or in the laboratory. When in their natural environment, organisms are often observed alive, whereas in the laboratory, they can be observed alive or dead. Organisms can be kept in the lab in special cases or vivaria, such as an aquarium for keeping aquatic animals or a wormery for keeping worms. Some organisms might be dried or preserved in a liquid. Samples may be collected and preserved for future study. For every organism or part of an organism, note the date, time, and place of observation. Write a brief description. Name the organism and draw a diagram or take a photograph. In biology, there are various equipment which are being used in observing organisms. All equipment in the laboratory must be handled with great care and according to the teacher's instructions or the instruction manual. During observation, only what is absolutely needed should be taken from the environment. Distraction should be avoided whenever possible. Preserved samples may be returned to the preservation liquid after observation. You can use the following equipment to observe organisms. A. Your own eyes. The human eye, which is responsible for sight, is the most precious instrument for observing organisms. Binoculars are also a good instrument or equipment used for observing faraway organisms such as birds. A hand lens or magnifying glass is also a good equipment used in observing, since it can be used to magnify organisms and make details clearer. Another equipment used in observation is a pair of tongs. Tongs are used for gripping and lifting organisms during observation. A petri dish is also a good equipment used for holding organisms during observation. Petri dishes are typically used in biology to observe cell growth, cell germination, and to study tiny animals. Another equipment used during observation is a thermometer. Thermometers are used for measuring temperature changes. Finally, Another equipment used in observing organisms is a pair of forceps. A pair of forceps are used for grasping and holding small objects which the fingers are too large to hold. Observation involves the careful study of many aspects of an organism. Appearance, for example, size, shape, and color. Structure, for example, the number and type of parts. Behavior. Habitat, that is where the organism lives. And population density, that is to mean, are there a lot of organisms or just one or two? Observation can be done in two ways, qualitatively and quantitatively. Qualitative observation makes use of the human senses such as seeing, touching, hearing, smelling, etc. These observations are descriptive. Quantitative observation uses both senses and instruments, such as a thermometer for measuring temperature, a ruler for measuring length, and a balance or scales for measuring mass. Qualitative observations are given a numerical value. Let us now examine what characterizes a good observation. A good observation should be relevant. This means that the observation should have a clearly defined objective. For example, the external structure of the organism, symmetry, orientation, or feeding habits. A good observation should also be accurate. This means that the description should be clear and written in direct and simple language so that any person reading it can easily find or identify the organism. For example, giving a description of where the organism lives, what it feeds on, how it reproduces, its external structure, or its shape. A good observation should also be repeatable. The observer should avoid making errors so that they make a similar observation another time or someone else could obtain similar results. It is also important to note the name of the organism being observed, the date of observation of the organism, the habitat or home of the organism, such as a lake, river, a marshy area, a tree, or a mountain top, the time of observation of the organism. Was it in the day or night, morning, evening? Was it a rainy or a dry season? It's important to note that. It's also important to note the weather conditions at the time of observation. In describing the external structure of an organism, it is necessary to take note of the following. 1. the orientation of the organism in space. The terms that are used to describe the orientation of an organism in space are: A. Anterior. This applies to the front end of the organism where the head is. B. Dorsal. This is the side of the organism that is normally directed upwards. It is often called the back of the organism. C. Posterior. This applies to the tail end of the organism where the anus is. And D. Ventral. This is a region that is normally directed downwards. It is often called the front of the organism. The picture below shows how the various terms used to describe the orientation of an organism and its features are being used. However, in humans, the terms are slightly different, as can also be seen in the picture below. Here, superior replaces dorsal and inferior replaces ventral. Another external structure to take note of is the organism's body symmetry. A line of symmetry is a plane along which an organism can be divided into equal and similar halves. Two types exist: bilateral symmetry, in which an organism can be divided into two equal and similar halves from the anterior to the posterior. If the animal is divided along this line from the anterior to the posterior, the right side R would be a mirror image of the left side L. Organisms that show bilateral symmetry include fish, birds, frogs, toads, lizards, and snakes. The seeds of some plants, such as groundnut and mango, and some flowers and leaves, also show bilateral symmetry. The second type of body symmetry is the radial symmetry. Here, the organism has many planes of symmetry. The picture below shows an example of radial symmetry. Lines A and A prime, B to B prime, and C, C prime are planes of symmetry along which the flower can be divided into two equal halves, each half being the mirror image of the other. The flower has many planes of symmetry. Many fruits, such as oranges, tomatoes, and pawpaw, and some animals, such as the sea anemone and the sea urchin, show radial symmetry. Other things to consider when describing the external structure of an organism include its color, general shape and size, its dimensions, that is in height, length, and width, and also any special structures that may be used for feeding reproduction, or movement. Let's now look at another special instrument used by scientists in observing organisms called the microscope. Microscopes are used to observe very small organisms that cannot be seen directly with the eye. There are several types of microscopes, such as the compound light microscope, as can be seen in the picture below, the electron microscope, and the dissecting microscope. The microscope has a system of lenses. These are arranged so that light is reflected by the mirror below the stage and passes through the organism being observed into the lenses. The image of the organism is enlarged or magnified to several times its original size. In this way, details can be seen and described. The table below shows the various parts of the compound light microscope and their functions. The first part is the eyepiece. The lens viewer looks through to see the specimen. The eyepiece usually contains a 10 times or 15 times power lens magnification. The second part is the body tube. The body tube connects the eyepiece to the objective lenses. The third part is the nose piece. The nose piece is a rotating turret that houses the objective lenses. The viewer spins the nose piece to select different objective lenses. The fourth part is the arm, and the arm connects the body tube to the base of the microscope. Next, we have the objective lenses. This is one of the most important parts of a compound microscope, as they are the lenses closest to the specimen. We also have the stage, which is the platform flat where the slide is placed. The seventh part is the stage clips. The stage clips are metal clips that hold the slide in place firmly. Next, we have the iris diaphragm. This adjusts the amount of light that reaches the specimen. The ninth part is the coarse adjustment, and it raises and lowers the stage for focusing. The 10th part is the fine adjustment. The fine adjustment fine-tunes the focus and increases the detail of the specimen by slightly moving the stage to sharpen the image. The 11th part is the light source, and it is a light source for a microscope. Older microscopes used mirrors to reflect light from an external source up through the bottom of the stage. However, most microscopes now use a low voltage bulb. Finally, we have the base, which supports the microscope, and it is where the light source is being located on the microscope. Very tiny organisms that cannot be seen with the eyes alone are called microscopic organisms. This means that they are organisms that can only be observed and studied with the aid of a microscope. A section is a thin, transparent slice of a part of an organism, or sometimes of a whole organism. There are two types of sections: longitudinal and transverse sections. A longitudinal section is made along the length of the organism, that is, from the anterior to the posterior, whereas a transverse section is taken across an organism. Sections are made in order to look at the internal parts of an organism using a microscope. Let's now look at how we can observe organisms in the laboratory. Organisms in the laboratory can be observed either alive or dead. Live organisms can be kept in vivaria or vivarium for singular. A vivarium is a specially prepared container for keeping living organisms. Examples of vivaria include an aquarium for keeping aquatic plants and animals, a snellery for keeping snails, and a wormery for keeping worms. Vivaria are useful because live organisms can be kept for a long period of time and can be studied often and in detail. Dead organisms are kept in the laboratory in preservation fluids. These fluids prevent the organisms from decomposing or rotting and keep them in a condition that is similar to when they were alive. Preservation fluids are also called fixatives. Examples of preservation fluids include formalin and alcohol. Examples of organisms that can be stored in preservation fluids include crabs, fish, snakes, cockroaches, snails, and lizards. Plant parts are easily dried for preservation. A plant press is used for this purpose. The observations and studies made of an organism must be reported carefully. Observations of biological interest can include any of the following: naming and classifying organisms that have similar features for easy study in the future, making drawings or biological diagrams of the whole or parts of an organism, making notes and illustrations in the form of nature calendars or observation charts, making microscope slides of the details as a long-term record, and taking photographs of the features as a long-term record. We have come to the end of our lesson. Let's now do a quick review on what we've learned so far from this lesson. In this lesson, we learned that organisms can be observed in the field or in the laboratory, dead or alive. It is important to avoid destruction when observing an organism. The time, date, and place of observation should be noted and the organism identified and named. Samples may be collected and preserved for proper study. Equipment must be handled properly and instructions respected. Some equipment used for observation include the human senses, a microscope, a hand lens, a thermometer, binoculars, preservation fluids, and Petri dishes. Organisms can be observed qualitatively and quantitatively. A good observation must be relevant, accurate, and repeatable. A description of the external structure of an organism should include the orientation, symmetry, the general shape and size, and any special features of note. In the laboratory, very tiny organisms are observed with the aid of a microscope. Sections of internal parts are also studied with the aid of a microscope. After careful study, the observation is recorded in several ways, including identifying, naming the organism, classifying the organism, making biological drawings or diagrams, making notes and illustrations, making slides, and taking photographs. To have a better understanding of our lesson, please head on to the revision questions that follow. Thanks for your kind attention.